Right now, as you watch this video, quantum mechanics is happening inside your body, not in some distant laboratory, not in a particle accelerator. Inside you, in your eyes, in your nose, in the cells of every living thing on this planet. And for most of the history of physics, scientists assumed this was impossible. The quantum world, they said, is the world of the very small, of isolated particles in perfect laboratory conditions, of temperatures near absolute zero. Biology is warm and wet and chaotic. Quantum effects, the story went, get destroyed instantly in biological systems. They cannot survive long enough to do anything useful. That story is wrong. And the evidence dismantling it has been building for 20 years. Welcome to quantum biology, the field that is quietly rewriting what we know about life itself. Let us start with your eyes, because this is the one that should stop you completely. When light enters your eye and hits the retina, a photon, a single quantum of light, collides with a molecule called retinal. And retinal does something extraordinary. It changes shape in less than 200 femtoseconds, which is 200 quadrillionths of a second, to give you a sense of how fast that is. A femtosecond is to a second what a second is to 32 million years. Nothing in classical chemistry can explain this speed. Molecular shape changes of this kind, in classical physics, should take thousands of times longer. The molecule should have to climb an energy barrier, step by step, like a ball rolling over a hill. And yet it doesn't. It moves almost instantaneously, with near-perfect efficiency. And the reason is quantum tunneling. In quantum mechanics, a particle does not have to go over a barrier. It can go through it. The probability wave of the particle, its quantum mechanical description, extends through the barrier. And there is a non-zero probability that the particle simply appears on the other side without having gone over. This is not a metaphor. This is a real physical process confirmed in experiments, and it appears to be happening in the very first step of human vision. Every time you see anything, every photon, every image, every face, every word on the screen, the first step is a quantum tunneling event happening in your retina, 200 femtoseconds after the light arrives. Let that sit for a moment. And then let us talk about the most celebrated example in quantum biology. Because while your eyes are extraordinary, the story of birds navigating using quantum entanglement is the one that launched the entire field. European robins migrate thousands of miles from Scandinavia to sub-Saharan Africa and back every year with astonishing precision. And for decades, nobody could explain how. They have a magnetic compass. We knew that, but the mystery was where. And how it was not in their beaks. The magnetic material there, iron particles, turned out to be in cells that have nothing to do with navigation. The answer, when it came, was stranger than anyone expected. It is in their eyes, specifically, in a protein in their retinas called cryptochrome. And cryptochrome works through a process that, when physicists first heard it proposed, many of them refused to believe it was real. When light hits cryptochrome, it creates what is called a radical pair. Two electrons in two separate molecules that are quantum entangled. Their spins, their quantum mechanical angular momentum are correlated. And now here is the critical part. The Earth's magnetic field influences how these entangled electron spins evolve. The spins precess, they rotate, and the rate and direction of that precession depends on the orientation of the molecule relative to the magnetic field. This changes the chemical reaction products, depending on which way the bird is facing, and those different chemical products create different signals in the bird's nervous system. The bird perceives the magnetic field as a visual sensation, a pattern of light and dark overlaid on its normal vision, like a heads-up display. Built from quantum entanglement in its eyes, this is not a theory. Multiple experiments with European robins have confirmed it. When you expose the birds to radio frequencies that disrupt quantum coherence, that scramble the entangled spins, the birds lose their ability to orient using the magnetic field. Their quantum compass breaks. Now we are going to go somewhere even more fundamental. Because what happens in bird eyes is remarkable. But what happens in every green leaf, in every blade of grass, in every photosynthesizing organism on the planet is even more extraordinary. Photosynthesis, the process by which plants and bacteria convert sunlight into chemical energy, is the most important chemical reaction in the history of life. Without it, almost nothing that lives today would exist. And for most of the 20th century, we thought we understood it reasonably well. We knew the components, the light harvesting complexes, the reaction centers, the electron transfer chains, but there was a problem. The efficiency was too high. When a photon is absorbed by a chlorophyll molecule in a light harvesting complex, the energy has to travel to a reaction center where it can be used, and it has to navigate a complex network of molecules to get there. Classically, this should be a hit or miss random walk. Energy bouncing from molecule to molecule, losing some with every hop. And yet, the efficiency is extraordinary, approaching 100% in some systems under ideal conditions. Almost no energy is lost. In 2007, Graham Fleming's group at Berkeley made a measurement that changed everything. They used ultrafast lasers, laser pulses lasting femtoseconds, to probe what was happening inside a photosynthetic complex from green sulfur bacteria. And they saw something that should not have been possible: quantum coherence. The energy was not hopping randomly from molecule to molecule. It was existing in a quantum superposition across multiple molecules simultaneously. And this superposition was allowing the energy to explore all possible paths to the reaction center at the same time. And then through a process related to quantum interference, the same principle that underlies quantum computing, the wrong paths cancel out and the right path amplifies. The energy finds the most efficient route, not by trial and error, but by quantum parallelism. The plant is running something like a quantum algorithm. Every time it captures a photon, the result, after 15 years of follow-up experiments, debate, replication, and refinement, is that quantum coherence plays a real role in the extraordinary efficiency of photosynthesis. Though the exact mechanism is still being worked out, and it is not just vision in photosynthesis, because now we come to the strangest one, the one that is most personal, and the one where the science is still most actively contested: your nose, the sense of smell. Works, according to the standard theory, by molecular shape recognition. An odorant molecule floats into your nose, binds to a receptor that matches its shape, like a key in a lock, and the receptor sends a signal. This is called the shape theory of olfaction. It is taught in every biology textbook, and it has a problem. Molecules that are mirror images of each other, the same atoms in the same arrangement but flipped, should smell identical under the shape theory, because they are the same shape, just reflected. But often they do not smell identical. They smell completely different. Carvone, for example. One mirror image smells like spearmint. The other smells like caraway seed, same molecular formula, same bond structure, mirror image shapes, completely different smells. The shape theory says this should not happen, and there is an alternative theory proposed by Luca Turin in 1996. That is much stranger. Turin suggested that smell receptors do not just match molecular shape, they detect molecular vibration. Every molecule vibrates at specific frequencies determined by its chemical bonds, like a tiny instrument playing a unique chord. And Turin proposed that the receptor detects this vibrational signature through quantum tunneling. An electron tunnels from the receptor to the odorant molecule, but only if the molecule's vibration matches the energy gap in the receptor. The molecule's shape gets it close enough that the vibration is what triggers the signal. The nose, Turin was saying, is a quantum spectrometer, not just a lock and key. This theory explains the mirror image problem immediately. Mirror images have the same shape, they fit the lock, but they have slightly different vibrational properties due to their different orientations in space. Different vibrational signatures, different smell. The evidence for the vibrational theory is real, but contested. Experiments with fruit flies by the group of Efthymios Skoulakis and colleagues in Athens showed that flies can distinguish between molecules with the same shape but different vibrational properties because the lighter isotope of hydrogen vibrates at a different frequency than the heavier deuterium. The flies could smell the difference between a molecule and its deuterated version. Same shape, different vibration. This is extraordinary evidence for the vibrational theory, but other experiments have pushback, and the field is still actively arguing. What is not argued is that the classical shape theory alone cannot explain all of olfaction. Something else is happening. Whether it is quantum tunneling vibration detection or some other mechanism, the lock and key picture is incomplete. Now let us zoom out, because the individual discoveries in vision, navigation, photosynthesis, and smell are each remarkable on their own. But together they are pointing at something much bigger. They are suggesting that life, in its 4 billion years of evolution, has learned to use quantum mechanics. Deliberately, that natural selection, in its patient, relentless search for efficiency, has discovered the quantum world and put it to work. Enzymes, the proteins that catalyze every chemical reaction in your body, appear to use quantum tunneling to transfer hydrogen atoms with speeds and efficiencies that classical chemistry cannot explain. DNA mutation, the process that drives evolution itself, may be influenced by quantum tunneling of protons within the DNA double helix, allowing mutations to occur in configurations that would not arise classically, meaning that quantum mechanics may have shaped the very history of life on Earth, by influencing which mutations were possible. And there is a more speculative frontier still, the question of whether the brain, whether consciousness itself, uses quantum mechanics in any meaningful way. The most famous proposal is the Penrose-Hameroff orchestrated objective reduction hypothesis, which suggests that quantum computations in structures called microtubules inside neurons play a role in consciousness. This idea is, to put it gently, controversial. Most neuroscientists regard it as speculative at best. The decoherence problem is severe. The brain is too warm and too noisy for quantum coherence to survive long enough to do computational work, according to most estimates. But the history of quantum biology, is a history of scientists saying quantum effects cannot survive in warm, wet biological systems, and then discovering that they do. The cryptochrome radical pair mechanism survives in the warm, wet interior of a bird's eye. Quantum coherence in photosynthesis survives long enough to route energy to reaction centers. The field has repeatedly proven that life has found ways around the decoherence problem, ways that we did not anticipate. So while Penrose Hameroff remains far outside the scientific consensus, the fact that life has surprised us repeatedly with its quantum capabilities means that the dismissal cannot be quite as confident as it once was. What quantum biology is ultimately telling us is something profound about the relationship between life and physics. For most of the 20th century, physics and biology developed in relative isolation. Physics studied the fundamental laws of the universe. Biology studied the messy, complex, warm, wet systems that lived in it. And the assumption was that the gap between them was large, that the quantum world was too strange and too fragile to have anything to do with the sprawling chaos of a living cell. That assumption is dissolving. Life, it turns out, did not evolve despite the quantum world. It evolved into it. It learned its rules, and it used them. 3.5 billion years of evolution is a very long time to figure out how to exploit the laws of physics. And what quantum biology is showing us is that life figured out quantum computing long before we did, that the plants on your windowsill are running quantum algorithms every time the sun shines on them, that the birds in the sky are reading the Earth's magnetic field through entangled electrons in their eyes, that your nose may be doing quantum spectroscopy every time you smell coffee in the morning, and that the very first moment light reaches your retina, and the world becomes visible to you as a quantum event. The boundary between the quantum world and the living world is not where we thought it was. It never was. Life was there first,